Okay, so let's take a look at problem 11.3, which is, let me read the question here. Compare conservative, semi-conservative, and dispersive modes of DNA replication. Okay, so what are those things? Let's okay, so let's start with let me see, best way to start this question. Okay, 11.3. So we are starting with okay, so Watson and Crick figured out the structure of double-stranded DNA, and one of the things that they noticed right away, and they even mentioned in the first report of finishing the structure or solving the structure, or at least their model, which turned out to be mostly true. And this is from our book. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So again, this was a quotation taken from the paper. And what does that mean? Let's take a look at just a little piece of DNA here. So let me diagram it like this. So this is one strand starting with the 5' end, and we have an A and a T and a C and a G, and we have the phosphates in between these nucleotides. Okay, well, forming the linkages between nucleotides. So this is one strand of DNA, and Watson and Crick's model, indicated that there'd be a second strand, and that strand would be complementary and anti-parallel to the first strand. So what do we mean by complementary? Well, everywhere we see an A on the first strand, we're going to see a T on the second strand. T on the first strand, A on the second strand, C on the first strand, G on the second strand, so on and so forth. So A pairs with T, 2 hydrogen bonds between those, 3 between C's and G's, and I can draw the phosphate backbone here that links the nucleotides together. And anti-parallel, so 5' carbon down there, 3' carbon up there, and we have essentially a diagram of double-stranded DNA. And what they were saying up here is that now that we have a model for what DNA looks like, we can say, oh wow, okay, you can immediately see how the DNA and how the genetic information can be copied and passed to the next generation. So what can happen? Well, we can just take this stuff right here. Oh, you know what? I'll move it over. Okay, here we go. Okay, so just take one strand, separate the strands, and okay, so this strand right here, this one up here now, let me switch to the pen, different color. This can serve as the template, to make one strand. And okay, that copies that strand, and this serves as the template to make the other strand. Okay, so DNA's been replicated. So they didn't know how DNA was replicated, but having the structure there right away suggested a very logical way that the DNA could be replicated. And as long as the replication process was careful, to make few mistakes, then you could copy the DNA and pass it to the next generation with few mutations. So this was very exciting. Okay, so like I was saying, very exciting. Now scientists want to figure out exactly how the DNA is replicated, assuming we're just going to split these strands here, and each strand is going to serve as the template for copying, for making a new strand. So there are a couple different ways that the DNA can be replicated with that sort of mechanism. And that's where conservative, semi-conservative, and dispersive modes of replication come in. So scientists were considering a number of possibilities. So conservative is one of them. So conservative replication. So how does that work? Well, okay. So here's one of the templates here. And I won't actually draw the bases. I'll just, draw it like this, and you can assume that, okay, well, this is an A, and this is a T, and this is a T, and this is an A. It doesn't really matter what the pairs are. Okay, we're starting with this, and for conservative replication, what happens, or what is thought to happen, if this DNA is replicated conservatively, the two DNA molecules that are produced would be composed of old strands or new strands. So here we go, the DNA was replicated, and at the finish, the completion of replication, when we look at those DNA molecules, we have one that's all old DNA and one that's all new DNA. New DNA. So both strands right here, one strand here, one strand over here, of the new DNA molecule are new DNA. And of the old DNA, both strands right here and right here, well, these are the original DNA molecules, and they're still together. And that's conservative replication. So two DNA molecules, one of them is comprised of two, both of the old DNA strands, and the other one is comprised of the two new DNA strands. So the next one that scientists were considering, they thought, well, maybe the DNA is replicated semi-conservatively. We switch back to yellow. Semi-conservative. So semi-conservative replication. So this is a mode of replication where you start with that old DNA here, 5 prime, 3 prime, 3 prime, 5 prime, and after replication, well, what do the two DNA molecules look like? Well, they look something like this. Okay, what am I doing here? What is this crazy professor doing? Well, what I'm doing is I'm showing you that of the two DNA molecules that are produced, after replication, each one of them is made up of a new strand of DNA and an old strand of DNA. So during the replication process, what probably happened is the DNA split, and this old strand right here, say, was replicated, and the new DNA, stayed hydrogen bonded to the molecule that the template that served that was used to replicate it. And the same thing over here. So this was the template, the old strand, and the new strand was made by copying this one over here, and they just stayed together. And this would be called semi-conservative replication, or the semi-conservative mode of replication, and it results in two DNA molecules made up of one strand of old DNA and one strand of new DNA. So one strand old, one strand new, one strand new, one strand old over here, but two DNA molecules. Okay, so that's semi-conservative replication. And we have one more that scientists were considering as a possibility. So let's take a look. Okay, this one is called the dispersive mode of replication. So dispersive replication, and this is probably the most confusing one. So I will draw our simple DNA molecule. I'm not going to put the helix in or anything. I haven't been doing that, and that would just be really messy. So with dispersive replication, what scientists thought might be possible is, depending on how replication happens, is you could end up with two DNA molecules that were made of strands that were a mix of old and new DNA. Okay, so I will just draw one of these here. So here's one strand. So you can see it has old DNA and new DNA here. And we scientists didn't know whether it would be like, it would go base by base with one old base, one new base, one old base, one new base, or nucleotide. But so it could look something like that, and that would be one strand made up of new DNA and old DNA. And over here we might have something like this. So it'd still be anti-parallel, right? And the bases would still be complementary. But okay, so here is a dispersively replicated molecule of DNA. And then we would just do the opposite over here. It's kind of fun to draw these. You could fast forward through this if you want to also, I'm sure. Okay, so we have two molecules of DNA. After replication, we had one over here, now we have two, and each of these is a mixture of, each strand in these is a mixture of old and new DNA. And this is the dispersive mode of replication. And it was Messelson and Stahl's experiment which really helped show which was the mode of replication that is actually used in organisms. So was it dispersive, semi-conservative, or conservative? And Messelson and Stahl's experiment is the one that most of us think that was the most important with showing which one of these modes happens in most organisms.